We're here at the 2018 CROI. We're here with Romas Geleziunas and with Gilead Sciences, and he is the man in charge of cure work. And we appreciate you taking the time you do every year, and it's always great to visit with you on where we are and some things that pop out as important. And we were just chatting about the work that Dan Baruch did. We did an interview with him, but I want your perspective as well on that. I think it's really important data.
Well, Fred, thank you again for having me. Always a pleasure to do our annual chat. And you know, I'm one of many people that are doing cure at Gilead, so we have a really fantastic team of people. So anyway, we're very fortunate to have this
You're the spokesperson, let's put it that way.
I speak about it more often than others? Exactly right,
exactly
right. Thanks for that clarification. Okay, so let's talk about this study because it's, I think, scientifically a very, very nice study. And I hear that you had Dan on this, presenting to your audience as well on this. So the general idea there, and we've been building on this theme when we've been talking over the years. We have this toll receptor 7 agonist that we've been studying for many years. In fact, almost 11 years since the inception of our cure program at Gilead. And we've been talking about pairing it with various agents. Last year when I came here to talk to you, I told you about a study that Dan Baruch and us and Janssen and the MHRP group did together. And we there combined the TLR7 agonist with the Janssen vaccine that's in clinical trials right now, the prophylactic vaccine, the same one, but with slightly different inserts. This particular vaccine was designed for SIV, right, to basically induce a response to SIV, the simian cousin of HIV. Right. But we did that study, and I told you about it, was TLR7 with the vaccine, and 3 of the 9 animals, so 33% of the animals, basically, after rebounding, after we stopped the ART in those animals, returned and went back to remission. So we had what appeared to be a functional cure in a third of the animals. We're very pleased with that result. So here, this is now the second installment, if you will. Now we're going to do, we did, the TLR7 agonist with a broadly neutralizing antibody. And you know this is a hot topic in the field because there's been a lot of great groups that are cloning these antibodies. And so the experiment that we did was simply asking if we combined a TLR7 agonist with a BNAb and treated monkeys that were ART suppressed, infected and ART suppressed, and we made sure that the drug and the antibody were there for quite a long time. Could we deplete the reservoir? Could we eliminate some of that reservoir? That was the question. So what's unique about this study is that the monkeys were suppressed for almost 2 years, which is unheard of in research, right? You know, so they were really, really well suppressed. And then the second piece that's really important that we did, which is a bit different than the rest of the field and how the field is pursuing these BNAbs, these broadly neutralizing antibodies, is when the last dose of the antibody was given, we waited for a long time to let the antibody wash out. So the antibody is gone when you stop the antiretroviral therapy. This is very important because those antibodies are also antivirals. Right. The antibody's there, you don't get rebound. It could simply be because the antibody is binding to viruses and you're preventing infection. Right. We did not want to answer that question. We wanted to see if we impacted the reservoir with that combination therapy, and the time to rebound would be an indication of that, right? The longer it would take to rebound, the more you would believe that something happened to the reservoir. So this was by design. So I think it was a really clever design, and I think it produced some really interesting results. I'm sure Dan told you about the results. So in the combination group, 5 of the 11 animals had not rebounded after stopping ART by 6 months. That's a pretty nice result. Placebo group, all 11 animals rebounded. So placebo was all entirely rebounding. And then the other 6 animals in the combination group rebounded and now are kind of trending down again. So we have a very nice result where it seems that we've done something to the reservoir, possibly diminished it to a point where now the animals are severely delayed in their rebound. So this is a very important result. And
it's exciting, and I don't know if we're so what would be the next step after this?
Good question. So the next step obviously is, this is a monkey study. Great, the science is beautiful. We're all very pleased with it, but it's got to work in people, right? So we've got to go find out. This is a scientific experiment that told us we should absolutely be testing TLR7 agonist and antibody in people. Right? So that's exactly what we're planning to do at Gilead. We have been going with the TLR7 program. It's called GS9620 for many years, slowly ascending the dose, making sure we find a safe, well-tolerated dose that gives us the biology that we're looking for. We also have an antibody called GS9722, which is the son of PGT121, the antibody that was used in Dan's study. And this is an antibody that has been enhanced in its functions and in certain capacities. So we have that antibody also now in phase 1 trials, so basically ascending doses in healthy volunteers. So the TLR7 and the antibody are running in parallel as monotherapies, but our goal is to combine them as soon as possible. We hope possibly by 2019 we're going to be able to put them together and test it in humans that are HIV infected and are expressed.
It'll be an exciting moment to see if it pans out.
Yeah.
It sure sounds like we're making some home runs. I know that I talked with Sharon, and she's done some presenting too here, and she had one of her colleagues.
This is Sharon Lewin, obviously.
Sharon Lewin, yeah,
good friend.
and she's doing a lot of work there. She was in the press conference when he was presenting. But I'm really excited about it. I think we're getting there, but I don't want to give anybody hope because it's way down the line.
Agreed.
But I think the one thing that we did realize, and you're kind of making evidence to it, is that it's going to take multiple treatments, kick and kill and all kinds of aspects of approach, but to maintain the non-detectability. And I don't know which is going to be a home run. It's just...
You're absolutely right, Fred.
And the one thing I always remind myself to ask or to tell is that this is a collaboration. You mentioned working with Janssen. I mean, you meet at meetings. We have sure there's integrity within the company and proprietary issues, but this is too big for any one of us, I think, to stand alone on. I think we share our data so that we all know kind of what's going on, and there are so that presents itself with opportunities to work together. And I guess things like, do you meet with other companies now while you're at the CROI?
Absolutely.
It's a great time. You're all here.
Exactly. We're all talking to each other. You're absolutely right. This is actually a theme that we started, you and I, many years ago. I was telling you how everybody's coming together. The climate has completely changed. It's not competitive. It's collaborative.
Right.
And it's very exciting to see, for example, the vaccine study with TLR7 that we talked about a few minutes ago. That's MHRP. That's U.S. Army government. You got 2 pharma companies and an academic group in Dan Baruch's team, right? Look at the combinations of players we're bringing to the table. And so I think that's very exciting, like you said, that we are able to gather resources from all kinds of spheres, put them together and work together, and look, we're generating ideas. And remember, this is all about generating ideas right now. What are the best ideas that we can move to the clinic? That's what it's about right now.
You got to stand back far enough to look at the big picture, I guess.
Exactly.
I've often said this, you can't see the forest through the trees sometimes, but
exactly.
I really, that is what always excited me about the move, and I don't know, Marty Delaney was the one that kind of always was interested in that collaborative spirit, and of course, you don't always see that, but of course now they have the Marty Delaney Collaboratories. Here's a good tribute to a guy who really felt strongly about all that, and always wanted to come out of a meeting with a win. Not that anyone would lose. We're talking about actually saying we have success, or that was a worthy meeting. If you go to a meeting, you come across the country and you don't get anything out of it, it's kind of a waste of time.
So in that spirit, for example, as you know, there are 6 Martin Delaney collaboratories right now, and we, Gilead, is involved with 3 of the 6 of them. So we're, again, the collaboration, we're involved with them, because together we'll find this, hopefully one day. We can't do this alone. No one can do this alone.
And the quicker the better, because we have, it just, as hard as every single interview that I conducted this time touched on the fact that people aren't doing enough. Community isn't doing it. There's so many ways that we can improve the prospect of reducing the transmission rate and the number of new infections. It's just there. I mean, we can do it, but it's a matter of people just not doing it. It takes people to make this happen.
That's right.
So we implore people, because we know that one day I'm sure we'll have a cure. I hope it's sooner than later. But meantime, people are going to get sick and do less well if they don't get on treatment. So it's important to do that.
Absolutely right.
So,
So just one more point. So we talked about the vaccine with TLR7 study that we talked about last year. This year, the antibody with TLR7. it begs the question, what if we combined all three of those, right? We got some remissions in the monkey model with the two combos. Well, could we do better with the three?
Right.
So this is a concept that obviously we've been very interested in for many years, and it will be tested in one of the collaboratories, one of the Delaney collaboratories that we're involved with. It's called the I4C, and Dan Baruch is the head of that with John Mellors and Nelson Michael from MHRP. So we're going to test that idea. We'll see if we can do better than the two pairs of agents, and if so, again, that brings us a new idea to go test in the clinic, and we become urgent if the data was positive. So I think we're doing the right thing and looking for the best ideas and then moving them to the clinic as fast as we can.
Well, the good thing is Gilead has a lot of revenue to do some good things with, and I know that you're doing that. That's really important for me to hear you doing the work and spending the bucks, because it's certainly well appreciated and I think well spent.
Well, in that regard, you know, we have this CURE program, CURE grant program that we talked about last year. We actually did a second version of it. We did a second round, if you will, last year, and we're going to do a third one this year. So this program, I told you, this is a philanthropic donation program. So we're basically identifying investigators, academic investigators that have great ideas and that we are basically funding, that we're offering them monies with no strings attached to do their research. So I know that the scientific community is very appreciative of this because it helps, and we're basically extending what we can do. We can't do everything, but others may be able to find, if something could help us find a cure, then so be it. We're funding that, so we're very proud to be able to help the academic community as well.
Well, again, we're really grateful for that. I don't know if there's anything more you want. Did you see anything at the conference that jumped out at you as worthy of tossing out in the audience here?
So the one debate, I know you're probably following this as well. So residual replication in people versus not, right? So this is a debate that's been going on for quite a while, and I always find it fascinating. And so we had a couple presentations, one by Mary Kearney and one by Thomas Rasmussen, kind of were addressing this. I find that the preponderance of the evidence suggests that we don't really have much replication in people that are taking their antiretroviral drugs consistently. There's not much evidence of that. So I'm not sure, that's an area that we should be pursuing. I'm a firm believer that I think we should go more after cure strategies, to try to eradicate this reservoir, rather than trying to imagine that we'll develop better drugs that will inhibit replication that I don't think is happening.
Right.
So this is a big debate, as you know. It's
I know John Kaufman years ago did the study, the viral load down to a half a copy or whatever that means. And I asked him, so how important is that? Well, not really, but we wanted to just get there. And so then I said, well, what is the number that makes a difference? I guess it depends on what you're going for. Yeah. But replication is, he said around 70 would be a good number as a cutoff. Well, we were at 50 early on, I think, when he was doing that. Yeah. And then we did investigative down to 20, and now I guess we're down in the small number of single digits. But so it's really not important. I think, like you say, maybe we should be just focused at the things that are going to have a what if not, and if it has something, if it's meaningful, then we need to know the answer.
Exactly.
Yeah.
So these questions, they basically reach the question of, well, what would happen if we were to further reduce, production of virions, for example? Well, in the end, I don't think that's going to lead us to where we want to be, which is prolonged periods of viral remission without ART, hopefully permanent one day. That's where we want to be.
Right.
Not adding another drug to reduce, viral load a little bit more. That's not going to change the game. You still have to take your antiretrovirals. I think we need to really...
Yeah, and it ain't going to make any difference whether you have a little bit of replication or not, and
exactly.
And it's inconsequential.
Exactly. So anyway, that debate is ongoing. As every conference, there's a debate on that topic, so it's not ending.
Well, I always enjoy meeting with you here, and we often have the community meetings where we can talk a little bit more science, and that's important. I think the people should realize that we're all human. We're all there. There was this issue back years ago that we're hiding the secret for AIDS, and where nobody is hiding anything.
No one's hiding anything.
Because I always bring up the subject. I said, how many people must be working at Gilead that are positive? They would be damn happy if they found a cure today. And they are working on the cure.
You know how hard we're working on this, Fred. We started, cure research at Gilead 11 years ago. I've spent the last 11 years of my life doing this. I mean, we're working hard. We're really doing our best to try to find.
And Gilead is a relatively new company.
Relatively new
compared to the old ones. Yeah,
exactly.
But I remember day one when they they came around, well, we don't have anything to sell right now, but we want to let you know we're here. And that was the early
days of antiretrovirals, exactly
Yeah,
right.
yeah.
So anyway, so we're trying to do our part, so we're working hard. So.
And thank you so much for that. Thank you.
Thank you.
All right, take care.